everyone. Welcome to our webinar, Microplastics Study Design, Analysis, and Reporting, Lessons Learned from our San Francisco Bay Study. First, I wanted to thank our funders, especially to Ocean Protection Council for funding this webinar, as well as the Gordon and Betty Moore Foundation for funding our original study, as well as additional supporters. During our webinar, if you have any questions, technical questions about our study or technical difficulties, please email us at sfmicroplastics@sfei.org. Don't use the chat function because we won't be monitoring that, but we will be monitoring the emails going to SF Microplastics throughout the webinar, and we'll be answering questions at the end. So we'll have 30 minutes at the end for that. This is our agenda for today. My name is Diana Lin, and I'm a senior scientist here at the San Francisco Estuary Institute, and I will start with an introduction. Liz Miller is an ecotoxicologist here at SFEI, and she will be talking about important factors in study design for microplastics. Rusty Holloman is a researcher at UC Davis, and he developed a transport model for our microplastic study for the San Francisco Bay, and he will be talking about microplastic data needs for the transport model. Then I will talk about field methods, and then we'll have a short break. We'll take a 5-minute short break. Then Chelsea Rockman, who is a professor at the University of Toronto, will be talking about the laboratory analytical methods used for our San Francisco Bay study. Adam Wong leads our data services team here at SFEI, and he will talk about data reporting. And then we will all be available at the end for questions and answers. So please email us questions. We'll have time to answer them. And joining us in answering questions is Rebecca Sutton, who was the lead scientist for the San Francisco Bay study, and Keenan Muno, who is a researcher in Chelsea Rockman's lab group, and she led much of the laboratory analysis for the San Francisco Bay study. The webinar is being recorded, and it will be publicly available and posted on our project website at sfei.org/projects/microplastics. This will be available to everyone, so you don't have to actually be a participant on this webinar to access the recording. So I'll start us off. So what are microparticles and microplastics? These are small particles that are operationally defined as everything less than 5 millimeters, as opposed to larger particles and litter items that are larger than 5 millimeters. In our study, we were very careful to be clear on our definition of microparticles and microplastics. Microparticles are particles that appear to be plastic and are smaller than that 5 millimeter dimension. Microplastics are microparticles that have been definitively determined to be plastic using analytical methods. And this is in recognition to the way the study of microplastics has evolved, where we realize that as we look at smaller and smaller particles, some of these small particles might be hard to distinguish whether they are plastic or not. Also in our study, we measured concentration of microplastics as a number of particles per volume of matrix. This is as opposed to mass per volume, which we might be more, we usually report concentrations in for other contaminants. This is based on the analytical methods that we used for measuring microplastic concentrations, and this is also more relevant for toxicity studies. Microplastics in the environment are very diverse. They come in various shapes and sizes, and we describe them based on their morphology. So they can look like fragments, fibers and fiber bundles, spheres, film, and foam. We also describe them based on other characteristics, like the polymer type of the plastic, additives that are included in the plastic, and the different plastics can also have different toxins that are absorbed to them. They have different colors and also come in a very large range in sizes. In our study, we made sure to describe the morphology and the polymer because this gives us an indication as to the source of the microparticles. For our San Francisco Bay study, this was a big collaboration between the San Francisco Estuary Institute, 5 gyres, and the University of Toronto. This was one of the first comprehensive regional studies of microplastics and was largely funded by the Gordon and Betty Moore Foundation. And the goal of the study was to inform pollution prevention action. We had many study objectives for the study, including improving methods for the study of microplastics, both in the field and in the laboratory. We also wanted to identify baseline conditions of microplastics in the bay and characterize pathways for how microplastics were entering the bay. We developed a transport model, and we also wanted to evaluate policy options based on the science that we were developing. And we also wanted to communicate our results widely. For our study, we were measuring a hazard, and here I wanted to emphasize the difference between hazard and risk. Hazard is something that has the potential to harm you, versus risk is the likelihood of that hazard causing harm. When we measured concentrations in the bay, we're measuring a hazard. We didn't do a risk assessment because we don't have sufficient understanding of the toxic effects of microplastics to develop toxicity thresholds. This is even more challenging to develop these toxicity thresholds when we consider the diversity of microplastics that we saw in the bay. We at SFEI will be collaborating with the Southern California Coastal Water Research Project to host a workshop to further our understanding of microplastic toxic effects later this year. So stay tuned. So for our San Francisco Bay study, we tried to do a comprehensive view by measuring various matrices. We measured microplastics in the surface water of the bay and the adjacent ocean, the sediment, fish, bivalves, as well as characterizing pollution pathways. And we measured treated wastewater effluent and untreated urban stormwater runoff, because in the Bay Area, most of our urban runoff is untreated. Our study results, as well as our methods, are fully described in our report, which is currently available on our website. There is a lessons learned chapter in the report, and we'll be sharing highlights in today's webinar. The bivalve study was a separately funded study, and so the report will be posted soon. We also have several manuscripts that are currently in preparation, including several manuscripts being prepared on the analytical results and evaluations. Also, a manuscript on the transport model is being developed, and we also will be having a lessons learned manuscript. And so you can reference these other publications for a fuller description of our lessons learned. And our goal today is to share some of our highlights in our lessons learned. We recognize that the field of microplastics is rapidly evolving, and we've learned a lot from our study. And so our goal is to share our lessons learned and share important factors that were important in our study, and also share some factors where we would do differently if we were going to do our study again. Since we're focusing on the lessons learned, our presentations will mostly be focusing on the methods instead of the results. And so you can look at our report for more of the results and findings of our study. Our hope is that these lessons learned will help guide you in your study of microplastics, and that together we can encourage robust and consistent monitoring of microplastics by sharing our lessons learned. Again, if you have any questions, please email us, and then I'll pass it on to Liz Miller.
All right. Hi, everyone. My name's Liz, and I will be discussing study design for microplastics monitoring in the environment. So just because there may be a wide variety of folks on the call, I wanted to start really simply with the basic what is study design, which is really thinking about how many samples we want to take and from where and when are we sampling. So this is also thinking about sampling within the constraints of having a budget and a timeline for your research, and really what kind of data you need to make good decisions in terms of best management to minimize microplastics entering the environment. So another way to think about this is that really what you want is samples that are as representative as possible for the state of microplastics in the environment. So I thought it would be useful to share an EPA resource for study design that many of you may not be aware of. So this is the Data Quality Objectives Process, or DQO's process. This is specifically a way to help researchers design environmental monitoring studies, and it's a seven-step planning approach based on the scientific method, and it helps you make sure you capture all of the possible things you should think about in designing a study before you begin your study. And you can find it at this link, but honestly, I think you could just Google EPA DQO and find it that way too. So the 7 steps are outlined here, but they're pretty vague and abstract from just the EPA definitions, so I'm going to go through them real quickly. So the first, to state the problem, is really understanding what's the issue you're addressing, in this case, microplastics in the environment, and what resources you have at your disposal in order to address the problem. So things like staff and budget and timing. Then you want to identify the decision, which is really actually thinking about the end process of your study, how your data will end up being used. And so in our case, we were planning on using our data to inform management practices to reduce microplastic pollution into the bay. Third is to identify the inputs, which means looking at the data that you have and the data that is missing and understanding what information is needed to best use that data. Fourth, define boundaries, which is when you start to really look at your specific site and understand what your focus is in terms of what matrices you're sampling, what is possible to do with your resources and with the state of understanding of the science, and really narrowing down what sites you should be sampling. Fifth is to develop a decision rule, which really means thinking about your analytical methods, what kind of measurements you'll be making, and then also thinking about your quality assurance and quality control, or QA/QC, how you're going to distinguish your measured values from contamination, for example. So 6, specify tolerable limits. This is thinking about the statistical analyses you're planning on doing and how you're going to recognize differences between samples. So, yeah. And then 7th, the final one, is to optimize the study design. So take those first 6 and then really finalize your plan. What are you actually going to do? So we did not actually use this framework for designing our study, but I am going to point back to the different steps when I talk about things that we think were really important in our study design. So one thing that is quite important and is easy to overlook is to have really clearly defined study objectives. This is not just in the abstract, but in the specific language that you're using. So you want to link to your overall goals, but you want to be concise, realistic, and using action verbs that are specific enough to actually be achievable. And then it's important to not change these objectives once your study has started. So an example from our study, when we started out, we had what I would consider a pretty vague objective of monitoring microplastics. Monitoring could really mean lots of different things, though. And then we wanted this to provide information on the contribution of microplastics to the ocean. So that's actually really more of 2 separate objectives. So a better way of stating this is that we wanted to determine a baseline for future monitoring. We wanted to understand the concentrations that are currently present so we can see changes into the future. And then we also wanted to investigate the contribution using our modeling approach. So thinking to number 4, defining boundaries, what sample sites you're actually using. There's some general considerations regardless of what matrix you're sampling that we think are important. Like I said earlier, making sure your samples are as representative as possible is important. And this could mean spatially representative. So for example, we wanted to represent the entire San Francisco Bay, not just one part of the bay. But also temporal distribution. So in California, we have the wet and dry season, and those can be very different. So we chose to sample during both to compare them. For microplastics in particular, we think it's important to understand your proximity to potential sources going into your system. So for us, that's why we chose to sample both wastewater and stormwater, because we thought those would be the main inputs of microplastics. And then at SFEI, we really like to have a less urban reference site, which allows us to compare kind of baseline levels without a ton of human impact to our highly human urbanized environment of the San Francisco Bay. So we use Tomales Bay, which is north of San Francisco Bay and has very little urbanization around it. So other things to consider is the type of sampling method that you're planning on doing. There's various types of sampling methods available for every matrix. And the reason this is important to think about right away is, one, logistically, but also the sieve size can really change the results that you get and how your data can be used. And I'll talk about that more in just a second. Other things to consider are things like ease of access. So what size boat do you need to get to a different sites may change based on where you're planning on sampling. Also things like whether you need a permit to sample in a certain location. If you're sampling multiple matrices like we did, it's important to think about how complementary your sampling sites can be so that you can directly compare. And then finally, we think it's important to have backup sites because when you're in the field sampling, sometimes things don't go as expected or you can't access a site that you thought you would be able to get to. So having a plan for ways to get around those issues is really important. Okay, so sieve size. This is an example figure from our report looking at wastewater effluent microplastics. And the main takeaway from this figure is that the size of the sieve that you're using to sample can drastically change the number of microparticles that you're seeing in your samples. And that seems to be more important than the location of the sampling or the type of wastewater treatment in this example. So the studies that had very small sieve sizes were able to capture many, many more particles than the larger sieve sizes, which let many particles slip through. So if you are looking to compare your data to other data in the literature or to other data you are planning on collecting, thinking about the sieve size and making those comparable is going to be really important when you're talking about the straight up numbers of microparticles in your samples. So like Diana said, we sampled a variety of different matrices in the bay, but thinking about specific site selection criteria, I wanted to focus in on our water samples. So this is both the surface water in the bay and adjacent ocean, and then also the creeks and streams leading into the bay, which we sampled during storms, so we're calling stormwater. So for those larger surface water bodies, we think it's important to include hydrology. And one thing that our study did really well is working with a modeler from the beginning to understand the hydrology of the bay and understand where there needed to be more data collected to really have a good understanding of what was happening in those more complicated areas. This could include also thinking about things like tides and currents, the wind speed and how much mixing there is at the surface, and then also more subtle things like whether there are convergence zones, lines with high concentrations, which Diana will talk about more in her next presentation. For smaller creeks and streams, we think it's very important to think about both your watershed size and the types of uses the land that's draining to the creek is that exists on the land that's draining to the creek. So in our study, we saw that watersheds that had high industrial land use had higher levels of microplastics, and so this is an important consideration when you're looking at where to sample. We also, like I said, sampled during wet weather, although most previous studies of creek and stream microplastic levels have actually occurred during dry weather. We thought it was important to sample during wet weather because there's evidence that rainstorms can actually mobilize microplastics from the landscape, and so that you're getting a big flush of microplastics during storms. But if you're sampling during storms, it's important to understand how much rainfall will increase the creek or stream flow above your baseline flow. So what size of a storm, how much rain you're getting at once should be defined ahead of time, so you know how this compares to your baseline non-wet weather. Storm sampling also has its own special set of safety considerations and logistics that are required, so it takes an extra bit of planning. Also, with creeks and streams, it's super important to think about your sampling methodology. These methods have not been as well developed as larger surface water methods, and things like mantitrals are not really practical to sample a small stream. The other thing about creeks and streams is you may want more than just the surface water. So we actually did depth integrated sampling, and once again, Diana will talk more about that in her next presentation. For quality assurance and quality control, a big part of this is blanks, both field blanks and laboratory blanks, which Diana and Chelsea will talk about more later. Thinking about how much contamination you expect will tell you how many blanks you need, and also how many are sufficient to distinguish between contamination and your actual environmental concentrations. Part of this is also thinking about your statistical analysis. How are you going to include your blanks in your analysis? There's not really a standard way of doing blank analysis in microplastics research right now, so there are a lot of different possible ways to do it based on what you think your data is going to be used for and how you collect your blanks. But QA/QC is not just about blanks, it's also about other assessments of data quality, including things like field duplicates, which can tell you how variable your sampling is at a single site, and then other things about thinking about how you can minimize errors. So for example, we saw a lot of orange fibers in our samples, and we linked them back to the orange PFDs we were wearing. So maybe if we had had newer PFDs, it would have been a different situation. All right, so one way that we think could really help you design QA/QC is to do a little pilot study for your field sampling. And we did this for our surface water and wastewater, but not our other matrices. And this can tell you how to refine your protocols to have the best possible results, and also can give you an expectation of the field concentrations you might expect. Because we had done our previous field sampling of surface water, we knew the levels we would expect there, but we hadn't previously sampled our stormwater, and so we thought that we would need larger samples than we probably could have gotten away with. We ended up seeing really large numbers of particles in those samples, and it maybe would have been good to take smaller samples and then need less time and effort in the lab analyzing them. Because of these sorts of things that we were running into during the study, we ended up subsampling. There's a lot of different types of subsampling, and it probably is a good idea to have a plan for subsampling, even if you don't think you will need it. It's better to have a plan and not use it than not have a plan and then need a plan. So a couple of examples of types of subsampling that may be necessary with microplastics analysis is how many locations you've sampled from, and then also the particle analysis itself. I have a couple of examples from our study. So all of the dots on this map are sediment samples that we collected, but we ended up running into a little bit of a time crunch at the end of our study because many of our samples had many more particles than anticipated, and so they took longer to analyze. And so we ended up having to choose fewer sediment sites to actually analyze. And this meant that it changed a little bit what we were able to do with this data. We've decided to focus on just the green dots, which are primarily near projected sources of microplastics, so primarily near wastewater treatment plant outfalls and stormwater inputs. And this means that we didn't necessarily have representative sediment samples from across the bay, but we did have an understanding of how sediment may be affected by these pathways. Another example is that many of our samples had thousands of particles, which is too many to do spectroscopic analysis on all of them. And so we ended up subsampling particles within each sample for our analysis to definitively determine the plastics. And Chelsea will talk about that more during her talk later. Another thing that's really important to think about in study design is your statistical analysis. Thinking about what kind of analysis you're planning on doing with your data can help you decide how many samples you need to be able to have enough statistical power to actually see differences. Also, understanding what kinds of significant differences you are expecting to actually consider differences, whether that's the standard P of less than 0.05, or whether you're thinking, oh, we only consider it different if there's an order of magnitude difference, for example. And I think it's really important to consult with professionals, really expert statisticians, because a lot of these analyses that we're starting to get into are much more complex than a simple t-test or ANOVA, for example. And then finally, I wanted to point out a couple of other possible pitfalls. We were really lucky in this study in that it was, from the beginning, a collaboration with a lot of different groups, scientists and advocates and policymakers and modelers. And this let us design a study that would give us the best possible data for addressing a wide variety of questions. Having backup plans, because there's always a possibility of things going wrong. So whether that's a subsampling plan or a backup site plan, that's really important to have. And then finally, in a project like this, working with a lot of different people, it's really important to have a record of all decisions that are made so that when there's changeover in staff or it's been 3 years, you know why a decision was made and how it was made. And that's it for me. Just a reminder, if you have questions or technical difficulties, the chat we are not monitoring, so please actually email sfmicroplastics@sfei.org instead, because we are monitoring that, and we will answer you if you email us. We will not answer you if you chat at us. And now I'm going to turn it over to Rusty, who's going to talk about modeling data needs.
Okay, hi, my name is Rusty Holloman, and I'm going to be talking about modeling data and what we need from the microplastic field data in order to adequately build transport models. So first off, just the general goals of the modeling portion of the project. One, we wanted to establish these mechanistic links between loads, namely stormwater and wastewater loads, to ambient concentrations of plastics in the bay, the ocean, and the sediments. Second, we want to know as much as possible where are microplastics going in the bay or in the coastal ocean, and so we can use the transport model to get a better sense, a richer idea of the ultimate fate of these plastics. Some of the modeling outputs are particularly useful for informing future monitoring, so we can see spatial patterns at finer scales, and maybe we can actually sample, at least at this kind of initial stage of the project, and use those to choose sort of the spatial and temporal distribution of sampling in the future. And finally, this is just there's a lot of new work going on in both the field and modeling side of microplastics, and we just want to know how well can the models perform, and are we at a point where models can be predictive, or is there still fundamental work that needs to be done to get to that point? So in terms of what we need from the field data in order to adequately model the system, for one, obviously we need some idea of the concentrations of the plastics that are coming into the bay, and those are just the fundamental inputs to our transport model. Second, we need concentration information from ambient water and potentially in the sediments as well, and that's such that we can take the model outputs and evaluate how well we're doing. Because these models without any, if you don't have the ability to compare your model output to observations, then we really can't constrain how skillful the models actually are. And third, and this turned out to be even more important than I think that we realized early on, was we need to know a fair bit about the actual particle characteristics, and in particular, have enough information about particles to get a sense of the distribution of settling and rising rates of the particles. Now, I'll go into that in greater depth in a few slides. So, the implications for field sampling. One, we made a point to sample near constrictions. So in the So I'm just gonna, here we go. So in particular, we made a point to sample surface concentrations, say, near the Golden Gate, and that was such that we can get a sort of a constricted flow and get a better overall estimate of how many plastics are moving from one portion of the bay to another. Like Liz mentioned earlier, we also were intentional in timing the sampling. In particular, we felt like the middle of the ebb or the late ebb or the middle to late flood tides were ideal because they're the least ambiguous. As you sample sort of close to slack or like just into the beginning of a tidal phase, then you may not be sure of whether you're sampling, say, water that was on its way out from the bay into the coastal ocean, or you may actually be sampling water that is just beginning to come back in. And so having samples that are timed and placed to be as, the least ambiguous as possible was at least our goal. We also made a point to sample both near and far from expected sources of plastics. Just anywhere where we felt like there could be some kind of gradient, we wanted to have enough data to get a sense of whether that gradient did or did not show up in the field data. And there were definitely some challenges along the way, and in particular with stormwater, for example, it was not always possible to locate the sampling sites, say these two stormwater sites in the far south, as close to the bay as we would like, and so that introduces some uncertainty about potential additional sources or sinks that we were not able to measure between that sample site and the open bay. And also, as Liz alluded to, the differing sieve sizes between the load data was mostly sieved at 125 microns versus the open water data that was sieved at 355 microns. That made direct comparisons between the model output and the field observations a bit more complicated. So getting a little bit into the particle characteristics and the vertical component of this. So we often think about particles just sort of mostly floating on the surface, but really there's a full continuum of particles that are either rising or sinking towards the bed or falling somewhere in between. For our study, we used the methods from a 2019 paper in ES&T. And that paper takes as inputs, the formulations from that paper take as inputs the density of the particles, the size, and some idea of the morphology, both in terms of roundness and the categories that Diana laid out early on. In particular, the density was based on the plastic type, which of course was coming from the spectroscopy and the lab analysis that Chelsea will talk about later on. All of these factors combine to give us some estimate of how these particles move up or down or roughly passive in the water column. To give you a sense of how broad the spectrum is, I'm showing here the distribution of those rising and sinking rates just from the wastewater load data. And this is just a histogram showing abundance ranging from highly buoyant floating particles on the left to the highly sinking particles on the far right. And this actually covers several orders of magnitude of that settling velocity. And in particular, I wanted to point to two features here. One is this peak on the slightly sinking side. These are primarily polyester fibers. And you compare that to a section of the buoyant side, polypropylene fibers. And as I'll show in two slides, the difference between slightly sinking and slightly buoyant is huge in terms of fate. But you could really only distinguish these particles if you did have that spectroscopy to actually understand what is the plastic polymer of each particular particle. And then I want to say a little bit more about the stormwater loads and the depth integration. So yeah, there was a lot of effort that went into fully depth integrating those samples, such that we get a full measure of particles that are near the surface as well as particles throughout the water column and near the bed. And you can imagine, based on that previous slide where we have such a distribution of particles that are some rising, some sinking, and actually are relatively few particles that are really close to passive. And so this is sort of exaggerated, but we see this kind of distribution out in the bay where the model at least suggests that the highest concentrations are actually kind of close to the bed and close to the surface. So it was definitely important to get the depth integration, and it's not enough to just say sample right in the middle of the water column, because you would probably be missing both of those peaks in that case. And so I've talked a lot about the sinking and floating, and so I wanted to show one bit of model output here just to drive home the point of how much of a difference it makes. So here I'm showing, these are both from model output during wet weather conditions. On the left, this is the distribution of particles that were slightly sinking. And you can see that they, for one, are really concentrated close to where they came into the model. And second, they're almost entirely within the bay. And you can compare that to the right panel showing the floating particles that are much more dispersed and are able to make it outside the bay as they float near the water's surface in much greater numbers. And so finally, just a few points of sort of future research. So we can see it's really important to understand this kind of rising and settling differentiation between particles. And there's some additional aspects that we were not able to get into related to the development of biofilms and how that can alter that characteristic of the particles. And so most of the research that's been done so far on establishing these rates has really used virgin plastics, often in the lab or in very controlled circumstances. And so I think there's a lot to be done using weathered particles, and in particular, particles that have been recovered through field sampling methods to get a better, more realistic idea of rising and settling in biofilm development. And another part that was particularly challenging in the modeling was understanding how these particles interact with the bed. And there's this kind of unknown, at least to us, balance between particles that are sinking and being transported along the bed versus particles that are sinking and actually joining the bed and no longer moving. So those are definitely 2 points where I think there's a lot to be done, and those would definitely improve the modeling capabilities. And with that, I will pass it on to Diana.
Here I just want to remind everyone that if you have any questions, please email us, and please don't use the chat function because we're not monitoring that. And don't press any of the orange bubbles on the top because some of you guys have been requesting presenters. So I'll be presenting the field methods that we use to collect our microplastic samples. And I'll especially be emphasizing how we collected our QA and QC samples because that's one of our major messages is that these are really important samples and it's really important that these QA/QC samples are representative. For our San Francisco Bay study, we collected a variety of matrices, and so I'm going to be talking about our field collection methods for each matrices, including the surface water samples, sediment samples, tissue samples, including the fish and bivalves, and pollution pathways, wastewater effluent samples, as well as our stormwater samples. So I have these numbers so that you can follow along as to how far along we are in the presentation. For our surface water samples, we used a manta trawl, which has a long history of use, which allowed us to compare our study results with other published studies. The manta trawl looks kind of like a manta ray, where it has an open mouth and it kind of skims the surface and collects particles that are floating on the water surface. And the net and the sieve size that we used was 1/3 of a millimeter. And so you can see in the photo that we rinsed all of the particles that were caught in the net into the end of the net, and then rinsed this into a sieve, and then ultimately put that into a sample jar. For our QA/QC samples, for our field blank, we made sure to rinse the whole net into the sieve and into the sample jar to represent the whole process of rinsing in the field blank sample. For the field duplicate, it wasn't feasible to pull 2 manta trawls alongside the boat at the same time, and so the duplicate was collected about 45 minutes after the first sample was collected along the same location. We also collected bulk grab samples, and for this we collected 1 liter sample to capture the smaller size fraction that wasn't captured in the manta trawl sample. Some lessons learned from our sampling effort was that the field lengths can vary significantly. Each time we go out, the conditions are very different, different people, sometimes different boat, wearing different clothes, and so that can affect what's in the field length. We also saw pretty significant variation in our field duplicate. For example, our relative percent differences that we saw range between 2 and over 100%. Liz earlier mentioned that it's really important to note foam lines and tidal fronts because these are locations that can trap vegetation, debris, and including microplastics. And so if you're pulling your manta trawl through these foam lines or tidal fronts, you can capture a much higher concentration than outside of these areas. So to address these challenges, we really recommend pilot testing to refine your method. For example, it can help you identify sources of contamination. From our initial field sampling and field blanks, we noticed sources of contamination. Note the black mat that's in the middle photo. We saw some black mat fibers in our field blanks, and we also noticed orange fibers that were coming from our flotation devices. And so pilot testing can help you identify potential sources that you might not have noticed before. And if you do find significant contamination, you can consider collecting different types of blanks to help identify the source. So you can collect different blanks representing different steps in your process. The pilot testing can also help inform the sample size needed for the sampling effort. So for our manta trawl, based on the contamination that we saw, our manta trawl samples were actually very large, and we saw lots of particles, which required a lot of time and resources for analysis. And so if we were to do it again for the San Francisco Bay study, we would actually shorten our trawl length. And then for our 1 liter grab samples, they were actually too small, and we would recommend, if we did it again, we would collect larger samples to kind of overcome some of the background contamination from the sample collection and processing process. And again, we recommend collecting more field blanks because given this variation, more field blanks will just give you more understanding and help you better represent that background contamination. Here is an image of the template that we use for the manta trawl samples to collect various field parameters. And so these field forms are actually available for download on our project website. Below the agenda, there's a link to downloading these forms. They're meant to be a starting point so that you can modify for your purposes. And I'll be highlighting in this presentation the most important parameters to record. And so if you wanted to simplify these templates, you could focus on some of these parameters. For the manta trawl sample, again, clearly the sample ID, the location, your start and end coordinates, as well as the date is very important. It's also important to record the sea state, and this describes the calmness or the turbulence of the water surface, and that can affect how much microparticles that are on the surface are getting mixed in those surface waters. We used a flow meter to measure the volume of water being sampled through the manta trawl, and so it's important to record the start and end flow meter readings. And we also recorded the boat speed and the current speed. And it was actually really helpful to kind of have these redundant measures of how much volume of water was sampled in the case of, say, like the flow meter got caught in some debris and wasn't turning correctly. And we noted the importance of noting foam lines and any other vegetation that you might be pulling the manta trawl through. Second matrix, sediment. For the sediment samples, we used a Van Veen grab and scoop samples from the surface sediment. And we collected from the top 5 centimeters because we wanted to represent current conditions of microplastic in the sediment. For our field blank, we actually rinsed all of the equipment with our milliQ water and rinsed that into a sample container to represent the field blank. And the field duplicate was collected using a second grab sample. We actually saw pretty significant contamination in our field blank sample, and our hypothesis for this is that it probably overrepresents the field blank contamination because for the field blank, we were rinsing all of our equipment surfaces versus for our field sample, we were actually very careful to avoid significant contact with other surfaces. And so really to address this, we really recommend collecting more field blanks. Another lesson learned was comparing our data results with other studies. Since there aren't standard methods for analyzing and reporting microplastics, sediment concentrations are reported in various units. And so we really recommend recording all of these different unit measures so that we can compare your data results with others and so others can compare as well. So this includes reporting wet weight, dry weight, volume, as well as the approximate surface area, which clearly you need to measure in the field. This was our template for sediment sampling. And these are some of the important parameters. And again, sea state is important. And to this template, I would actually recommend including the approximate surface area and volume of the sample collected. For tissue sampling, we actually did not collect a field blank because there aren't really any good methods for collecting a field blank samples for these tissue samples. And for the fish sampling and bivalve sampling effort, we actually took different sampling and compositing approaches. For the fish samples, for each of our sampling locations, which you can see a map figure of on the slide, we took 10 individuals per species per site. And for the bivalve study, we actually took a different result and composited bivalves for samples and took 3 composites per site. And so depending on your sampling budget and the goal of your study, you might take different approaches to how you sample. Some lessons learned from the sampling is that we saw a pretty significant variation in the particle counts per individual, which can affect your data analysis. This graph shows the particle counts per anchovy from our San Francisco-based study, and you can see that the distribution of particles is pretty skewed. There's a few outliers where there are much higher concentrations versus most of the samples had lower concentrations. We also noticed that it's really important to be transparent about the fiber counts, first because there's significantly more fibers than the other particle types, and also many studies don't report fibers, and so it's really important that users can either include or not include the fiber counts from the total particle counts. And while we didn't have field blanks for these tissue samples, having the lab blanks was really important for representing some of the background contamination that might occur from processing these samples. For wastewater sampling, we collected final treated wastewater effluent through a set of sieves. And for wastewater sampling, we actually added the 125 micrometer sieve in order to capture a smaller size fraction. In this photo, this was one where we actually added another 1 millimeter sieve to capture any large particles, and that just helps prevent some clogging of the smaller sieve sizes underneath. We actually designed this upside-down bucket with a flow meter that we placed on top of the sieves to reduce any contamination of the sieves by kind of protecting it from air deposition. In order to reduce variability in the sampling, in order to reduce the variability in our sampling, we took 24-hour samples, so the effluent flow was allowed to flow for a period of 24 hours, and this was in contrast to our previous study where we only took sampling during peak hour flows, and so this was to make sure our sample was more representative of flows. We also collected our samples during weekdays. Our sampling design was to sample at facilities with secondary treatment as well as facilities with an additional advanced filtration. And for our field blanks, we did this by putting an extra set of sieves in the same sampling location using a similar setup, and then rinsing that blank sieve into the sample container for the field blank. And for the field duplicate, we were able to set it up with this Y splitter so that we could collect the field duplicate at the same time. So some lessons learned from the sampling. These sieves are set out and usually sampling a lot of water over 24 hours, and the sieves can get clogged, especially considering what type of treatment process is happening at the wastewater treatment plant. And so depending on how much particulate and other algae growth might be happening in the effluent, the sieve can get clogged when unattended, and so the way to address this is either to reduce your flows or to switch out the sieves halfway through your sampling effort. Also, the conditions at each facility can be variable, and so when you're collecting your field blanks, you want to think whether that's representative of each facility. And really, the way to address this is to collect more field blanks, and if possible, to collect a field blank at each facility. This was the form that we used for collecting the wastewater sample in the field. Again, the date and start-end times, measuring the amount of volume of water that was sampled through the sieves over the 24-hour period, and also marking the sampling location and where in the treatment process. I actually found it really important to record this when you were in the field, because then I was able to ask the plant personnel exactly where, just to confirm where we were sampling from the treatment process. For example, at one sampling port, there was one faucet that was sampling dechlorinated and another faucet that was sampling dechlorinated. And so it's helpful to be very clear as to what you're sampling when you're in the field. Last matrix for stormwater sampling, we used a similar set of sieves for the stormwater sample. And we used an ISCO pump to sample from the creek or storm drain or river that we were sampling from. And the way we sampled was to collect, use the ISCO pump to take sips through the entire storm. So, for example, if it was a 4-hour storm, we might take a sip every half hour. And we moved the end of the pump tube up and down in the water column to get that depth integration, which was really important for our study because we wanted to do a load calculation. For the field blank, we just had a second set of sieves that was exposed to the air while we were collecting the sample. And when water was not being sampled, we had the sieves covered with aluminum foil. For the field duplicate, we had a second set of sieves and took the sip right after the first sip. And so we were collecting the duplicate right afterwards. Some lessons learned from the sampling effort. We found that depth integration and capturing the full storm was really important. There was one case where we weren't able to do depth integration and weren't able to capture the full storm because we weren't quite ready at the beginning of the storm, and these samples had lower concentrations than we would have expected. We also saw pretty high concentrations, and we got a lot of particles in our sample. And so if we were to do this again, we would probably reduce our sample size to reduce the resources needed to analyze these samples. Another interesting finding from our study was that about half of the particles were these black rubbery fragments. And these particles actually needed a different method of analysis. So for most of our particles, Chelsea will talk later, they use mostly Raman and FTIR spectroscopy for analyzing these samples, and these black rubbery fragments were really hard to analyze using spectroscopy and require a different analytical methods for analysis, such as pyrolysis GCMS. And while there are various potential sources of these rubbery particles, tire wear is a potential source. And currently, as we're doing more research as to tire wear particles, we're finding that tire wear particles are probably, most of them are actually smaller than our smallest sieve size. And so if we wanted to go back and better quantify tire wear particles, we would want to use a smaller sieve size. And so this is really important to the amount and type of particles that you might be sampling. This is the stormwater field sampling form that we used, and we actually used the same form for collecting samples for a variety of other contaminants. And this is a fairly simple form where we just record the start and end time of each aliquot or each of the sips that we're taking during the sampling. So some key takeaways. QA/QC samples are really important. It's really important to minimize contamination in the field, and you might do this by doing some pilot studies to identify any potential sources of contamination. It's also important to carefully design field and laboratory templates to make sure that your important parameters are recorded. And it's also helpful to simplify these to reduce any burden and confusion in the field. The results of our full study are published on our project website, and so the findings and results you can find there, which is why we're mostly focusing on the methods of our study. And so if you have any questions, please email us. Again, please don't use the chat function because we're not monitoring the chat, we're monitoring this email. And now we'll be taking a 5-minute break.
I'm going to talk today about the methods specifically that are related to the lab. And the one thing I want to note is that I am Chelsea Ruckman that's doing the presenting, but I'm presenting with Keenan Munot, who was introduced in the beginning, and also the work of more than 20 students and staff that were in the lab, because a lot of plastics means you need a lot of people to do the work. So we can go to the next slide. So I just want to start by just pointing out the matrix that we're talking about here, or the analyte that we're talking about here. So microplastics are not just one thing. There's lots of different types of polymers, they come from different product types. There's different chemistries, different sizes, things that Diana pointed out in the beginning. In our lab, we were interested in identifying the polymer type, the size, the shape, and the color of all of the particles that were in our samples. So going on to the next slide. So I'm going to talk today about some of the best practices in the lab, some of which we had in practice before this project, and some of which we learned by doing this project. So I'll talk about the sample preparation, the analysis, and the data reporting. Next slide. So a lot of what we used in this study in terms of best practices actually come from the field of analytical chemistry. So in many ways, microplastics, as I said earlier, are just another analyte. So for sample preparation, we wanted to have clean laboratory practices. We always had blanks that we used in the laboratory that were mostly RO water going through the same process as the samples. And then we tried to reduce the amount of plastic that was used in our lab, including through our lab coats, and then as much as we could through our lab equipment. For the sample analysis, chemical identification of the material is really important, and that's more important as you get into smaller particles. It can confirm that what you're looking at is actually plastic and then give you some idea of what the material is. For this, we used Raman and FTIR spectroscopy, and then, as Diana mentioned, pyrolysis GCMS for some of the rubber particles. For data reporting, it can be really important to report the recovery of the extraction method that you're using. The limit of detection or limit of quantification, that's really mostly related to size for microplastics. What size can your method see down to? And then blank subtraction. If you find particles in your lab blanks or your field blanks, how do you subtract them from the samples? In this study, most of the data was all kept there visible for people to decide how they wanted to deal with that data to be conservative. Other labs may subtract by color morphology, for example, or even polymer type. Next slide. So just to go over in detail some of the clean techniques that we use in our lab, dust is our worst enemy. We wipe our bench tops regularly. It's important to have your floors mopped. We do all of our work in a clean cabinet when we can, so filtering and other things where we can avoid dust exposure completely in the sample. When we can't, we do have a HEPA filter in the lab, which reduces the dust in the air, and we cover our samples whenever possible. We also wear cotton lab coats, we clean our tools with water between use, and we use glass and non-plastic materials where possible. It's also important to be aware of and catalog the type of contamination you might see in your samples. So for example, Liz mentioned those orange fibers from the life jackets. So because we knew that those were in the field blank, we knew that when we saw them in a sample, they probably weren't coming from the environment. So next slide. It's also really important to have strict training protocols. So as I said in the beginning, there were a lot of different students and researchers working on this project. And in an ideal world, you might have just one person counting one particular matrix or all of the samples for the whole study. But in a project of 400 plus samples with thousands of particles and altogether hundreds of thousands of particles, you have to have multiple people. So we have developed a very strict lab training protocol where we have SOPs or standard operating procedures for every matrix. We have a color key, we have a morphology key, which I'll talk about in a bit. And then we teach people how to use touch and feel in addition to visual when they're under the microscope. And Keenan has created like a plate of microplastics where she knows how she would categorize them. Some are plastic and some are not, and basically to test people at the end of their training. And it's important to do this protocol and also to consider people bias and maybe do some statistical analyses to see whether or not you have that bias to figure out and inform how you can inform that bias. Next slide. So I mentioned earlier a morphology key and a color key, and I'm not going to go through these in a lot of detail, but I just want to show you some pictures of what we're talking about when we talk about these different morphologies or shapes. In the San Francisco Estuary Institute study, we looked at fragments, spheres, pellets, fibers, fiber bundle, foam, and film. But we put pellets with spheres because we really didn't actually have very many pre-production pellets in this study. But these are the 7 categories that we use in our lab. And these morphologies or shapes can often tell you something about the source. So we think it's important to consider these. So if you go to the next slide, I'm just kind of going to quickly go through a series of photos to show you what these different morphologies look like. So for fragments, just gives you an idea of what types of different fragments you'd see. The rubbery fragments from the tires or the tire and road wear particles would fall in here. Next slide. Just an example of some of the different films that you see, the different colors and the different textures. Next slide. The different types of foam. I think a lot of people think about styrofoam when they think about foam, but to note that we see foam from construction and foam from other types of materials, maybe polyurethane. Next slide. And then we also see spheres of different sizes. A lot of people might think about microbeads when they think of these. We also saw perfectly spherical glass beads that we think come from road paint. Next slide. And then pellets. And I apologize for these slides on my screen because I have a Mac. It looks fine, but I think we've got a Mac PC issue, so just know that that says pellets. Pellets are one of the easiest things, I guess, to distinguish what source they came from. These are pre-production pellets from industry. Some people call them nurdles, and they come in pretty distinct shapes. So often they're cylindrical or oval-like in the 3 to 5 millimeter range. Next slide. And then for fibers, and then if you go on to the next one, you have fiber bundles. We consider these 2 categories differently in that fibers are one individual fiber, but the fiber bundles is kind of a mess of lots of similarly colored fibers together that can't be broken apart, that may have entered the water as kind of that fiber bundle pill from clothing, as opposed to being like lots of single different unique individually colored fibers that you might see in kind of the mucus layer in the ocean. So we make sure that when we call it a fiber bundle, it is all a consistent color. Next slide. And then we also have on our morphology key in our lab lots of examples of natural materials that might be mistaken for microplastics. And this is a useful tool for students to look at when they're seeing something under the microscope that might be fishy. Next slide. We also developed a strict color key after going through this study. So one of the things that became apparent really quick is how different people might categorize the color of different particles. And so if you click one more time, you'll see some of the examples of some of the pesky colors that ended up in our data set, like light blue versus dark blue, or magenta or teal, which you could easily have maybe called pink or a blue or green. And so we have a color key where we only use these specific colors, and it's really important to make sure that they're either all capitalized or all lowercase, just to standardize your lab for your data set. Next slide. Then we mentioned that we have different standard operating procedures, and we have measured the recovery of every single one of those extraction protocols to make sure that we feel good about the extraction or the recovery of each of our extractions. So we aim for more than 80% recovery. The way that we do this is we create a matrix in our lab. So if it's fish, we'll just buy fish from the market and cut up a fillet and spike it with microplastics. And if it's stormwater, we get some RO water and add some dirt and leaves from outside. And then we add plastics of a known shape, polymer type, and color of a known count. And we usually do 4 or 5 different types. And then we do the extraction and we look at our recovery. And then once we feel good about that, we proceed using that method. Next slide. So to talk about some of these different extraction methods, and we have these standard operating procedures on our computer, and we don't actually mind if someone sends us an email and asks us for them. So if you go to the next slide, I'm just going to kind of go through what these different procedures are. So this does not include the sampling because that was already talked about from Diana. It's just going to go from the extraction, the identification and categorization, pictures and measurements, and chemical analysis. The next slide. The one thing I do want to say is that we size fraction in our lab. We think this is important for a few reasons. When we are doing extractions, we extract we size fraction pretty early on in the process. It reduces the particle load in each individual sample for counting and picking in terms of in that size range. It creates bins for data analysis. Let's say you don't have the manpower to take pictures and measurements of every particle in your sample. You have binned them with your sieve size so you know what size range it is. This can be troublesome for fibers, but for fragments it works well. It's easier to focus on similarly sized particles under the microscope, so it makes counting more streamlined. And you also avoid particle overlap because you have less particles in your sample. And then, so now if you go to the next slide, I'm just going to go through each of the different extraction protocols that we used. So for surface water samples, this includes both Manta as well as 1 liter. We actually used a really simple procedure, which was just simply sieving and filtering to a smaller size range for the 1 liter sample. I know that some people use a wet peroxide oxidation for Manta sampling, and that for some samples, especially in freshwater, we find that's really necessary. For these samples, we decided not to do it because we didn't have a ton of plant material in our samples. There were only a few samples that were pesky where we felt like that might have been useful. So all we're doing is sieving or filtering and then separating the sample into different size jars with different size fractions. Next slide. For sediment and the stormwater samples, we use density separation. This includes using a hyper salt solution, so a dense solution that's denser than the microplastics, to float the plastics to the surface and sink the sediments down to the bottom. In our lab, we use calcium chloride. We do this because it's less expensive and safe. Some people use more toxic chemicals or more expensive chemicals, but we were happy with our recovery with this method, and so we proceeded with using it. And then, of course, we size fraction when we're done with that density separation. Next slide. For fish and wastewater samples, this is where we use a chemical digestion. So we use potassium hydroxide, or KOH, at 20% in RO water. We use a low heat to let it bake, sometimes overnight, sometimes it takes a week to get rid of the organic material. And then we do our sieving just to the smallest sieve size you want to get rid of that potassium hydroxide. And then we add a detergent to get rid of some of the fat and break it up a bit. And then we do our density or our size separations with sieves, and then we have our samples ready for counting. I think that's our last extraction. So the next slide is how we picked, and this is why we have so many students. So we had lots of different samples that needed to be sorted visually under a microscope. For some labs, they might have automated techniques using Raman or FTIR, but these are pretty novel, and I would say there's a very small percentage of labs that have automated counting and analysis, and the majority are using this particle picking method. And I would say that for large particles, this particle picking method is superior, no matter what types of materials you may, what types of instruments you may have. It's practical. So first, we wipe down all of our surfaces for good QA/QC, put on our cotton lab coats, cover our beakers, and then we collect all of our materials, which you can see here on the side. If you go to the next slide, you can just see what our workstation looks like. So here is just a microscope with all of the different tools that we will use. And then if you go to the next slide, you can kind of see our process. So we have our jar of a certain size fraction. We take a spoonful from the jar into a counting dish, rinse the spoon to make sure we have everything, identify the particles moving through using a grid and a black microscope base. We pick any suspected particles and place them in a particle dish. And then we repeat identification and picking using the white microscope base so that we can see the darker particles. And then when we're done, we pour the contents of that counting dish into a clean beaker. That now becomes waste, and we take a new spoonful. And we do that until the jar is empty. And then we switch to the next size fraction. So this is laborious. Go to the next slide. When we are picking the plastic and putting it into a counting, or into a, not the counting dish, but the particle dish, in the beginning of this study, we were actually picking every single particle, and we tend to mount them on double-sided sticky tape. Now, that is because we are prepping them for Raman spectroscopy, and for our instrument, that's the best presentation method that we have, besides a filter. And in many cases, this mounting works really well because it keeps the fibers steady, for example. But this is really laborious. So we ended up doing a subsampling of our picking. And now in our lab, what we do is we pick and label on the sticky tape the first 10 of each color morphology. So the first 10 black fibers, the first 10 blue spheres, for example, from each size fraction. Next slide. And this just shows you an example of what it looks like. They're actually quite pretty. And then these plates can actually go straight under the ramen. We actually use projector paper that we cut into a circle, which you can't see in there on the bottom of the dish. So you can actually remove it and put it straight under your ramen microscope. Next slide. Then for all of those picked particles, we take pictures and measurements of all of the different particles, and we do this with ImageJ. You can do this with any software that you have that might be linked up to the pictures on your, or the camera on your microscope. Just be careful with certain things. You don't need to measure the frayed projections and fibers, and you can use segmented lines or curved lines when necessary. And then if particles are broken, you have to use judgment to take measurements of the different pieces. Next slide. So then finally, I'm going to talk about the chemical analysis. Now, there's two reasons why this is important, and it depends on the type of chemical analysis that you are doing. If you are doing an automated analysis and you're looking at small particles on a filter, you're going to be getting the chemical characterization of every single particle. So now you know exactly what particles of what you picked are anthropogenic, what types of materials they are. But if you're subsampling, then this chemical analysis also gives you a better, better confidence over your subsampling method. So it helps you know how well you're doing when you're picking what you suspect are anthropogenic particles. So we in this study used Raman spectroscopy and FTIR. As I mentioned, we did use some pyrolysis GCMS for those black rubbery particles, just to have a good idea of what some of those were. So if you go to the next slide, you can just see what some of the data looks like. This is just an example, but because we were subsampling and not identifying every particle in our samples, I just want to show you that 4% of the particles that were picked in this example were not anthropogenic, and 7% were unknown. So you can say, okay, 89% of what we studied here we think are actually anthropogenic. 58% of them gave us some sort of plastic polymer. We knew the polymer distribution in our sample. Then you're looking at this and saying, what in the world is anthropogenic unknown? So if you go to the next slide, this is just an issue that we deal with our Raman spectrometer that I think sometimes you also get with FTIR for different reasons, and sometimes with GCMS, where you can't quite figure out what the polymer is because it's being masked by a dye or an additive, or you get fluorescence, for example. With Raman, now if you have fluorescence, you're going to get an unknown, but if you get a dye or an additive, you know that it's anthropogenic. You just don't necessarily know that underlying material. So we had these different categories of how we discussed them, whether it was anthropogenic, a dye, but we didn't know what it was, whether it was plastic or cotton. We have anthropogenic cellulosic if we knew that it was a cotton, and synthetic if we knew that it was like a plastic additive, but we weren't quite sure what the material was. So this frustrated us, so we took advantage of this study to create a better method. So you go to the next slide. So Keenan and then also Hannah in the lab worked really hard at building Raman libraries with the particles that we had from this study. So first we just collected different plastics off the shelf to make a Raman spectral library that looked more like plastic. So instead of being like clean virgin plastic standards in the library, we bought colorful plastics that would have additives and different morphologies or different flexibilities. But then Keenan took a lot of those particles from the SFEI study and other studies from the environment, which might be oxidized or have biofouling on them, and added those to the library too. When we knew what they were, we could confirm by Raman and FTIR. We put them into a library that we call SLOP, the Spectral Library of Plastic Particles. What I'm showing you here is how much it actually helped fix some of this problem. So the first bar on the left is the original match before we had our libraries. That blue being coming across as anthropogenic unknown. So here you have several particles that are coming out as anthropogenic unknown. And then after we run these particles, and this is, I think, over 60 particles, quite a few, so somewhere between 60 and 100, then you can see that the anthropogenic unknown shrinks substantially, and all of a sudden you have a better idea of what those materials are. So this was an extra bonus that we learned, lessons learned from this project. Go to the next slide. Just going to talk, this is my final slide, is just talk about some of the best practices for reporting. So now you've done your extractions, you've done your chemical ID, you have all your data. You want to make sure that when you're writing this up, you describe that QA/QC as best you can in your methods. Reviewers are absolutely looking to make sure you have clean techniques, that you have blanks, how you subtract them, what the recovery of your method is, and if you had a subsampling strategy, reporting what it is. When you show your raw data, it's important to give the particle size, the color, the morphology, and the material type. I think the color may be less important, but a lot of people do want to see that data. And then give the plastic concentration and number of particles per unit volume. But then some people would like to see weight too, if you can do it. We often don't because it's not easy to weigh microplastics. And then we really suggest sharing all of your data for synthesis studies. So of course, we're not asking you to share all your data before it's published, but once it's published, we feel strongly that it's useful to put your data sheets in your supplementary or upload them on some sort of online data repository where people can use them for big synthesis studies or to compare with their studies in different places. And with that, you can go to the next slide, and I will say thank you and apologies for the late start, and we look forward to answering questions later.
Oh, no.
Oh.
Hi, I'm Adam Wong. I led the data management effort for this project, and I'm going to give sort of an overview of the process of getting data ready for analysis and public release after we've received it from field crews and Chelsea's lab. So we had some challenges in that we were receiving data over many months across lots of different matrices, from just one lab, but lots of different field crews in many different submission templates. And so standardizing all that sort of information and then also collecting extra metadata, just that's a process that I'll go through. And then we're preparing data for upload to several different online tools. With the help of the Ocean Protection Council, we are uploading to the California Natural Resource Agency's Open Data Portal, the Environmental Data Initiative EDI portal, and CEDIN, which is the California Environmental Data Exchange Network. So some of the stuff I'm going to go into might seem a little bit in the weeds, but as other people have described, microparticles can be a little bit more complicated than a traditional contaminant, and they have lots of different ways in which they're characterized as opposed to just a simple concentration. And each of those different characteristics needs to be standardized in such a way that they can be either aggregated or analyzed for the whole project. And as Chelsea mentioned, you know, if you were doing a solo project where you had just one person doing all the analysis, it's a lot easier to maintain consistent, like perfect consistency across all of the analysis. And sort of similarly, with all the different matrices, we were dealing with many different field crews, and that meant challenges in data entry on the field sheets and stuff as well. I'll discuss some of the caveats that are important to consider for our data, but it's also about thinking through a future study design, some caveats that you want to be aware of and that you will need to plan to address even in just the study design phase. So we get in the field data in field data sheets, as Diana showed, primarily they're being reported to us on paper. You could probably do some of it on a computer, but the reality of field sampling, especially with something like stormwater, means that you're going to have people writing on paper, and so you have to digitize that information, and that's not just, you know, scanning and making a PDF. It's recording data into spreadsheets and tables so that they can be stored and combined with your analytical data later. Part of this process is correcting errors. You'll have typos either in the handwritten information or in data entry. It's also truth checking things like coordinates, mapping them, making sure that they make sense, that they match up with where samples were actually taken. And then there are some standardizing issues where you have dates and different codes that represent pieces of collection information that may not have all come in exactly the same format, and that's again part of the situation when you have so many different matrices, so many different field crews, stuff like that. So eventually we get everything standardized, stored in a database at SFEI. It provides us an opportunity to add calculated fields, so that can be stuff like adding in days of the week or metadata about different station codes, things like that. It also provides an opportunity to record information about the subsampling decisions that are made, things like broken bottles, or if there's limitations on time and funding. You may have, as Liz pointed out, you only are able to analyze samples from a subset of your stations. You want to record that information so that when you're getting data from the lab, you know that you're not missing anything. And it also is an opportunity to store information if you, as part of your study design, had developed contingency plans ahead of time. It's not something that we were able to do, but this sort of step is where you could store that information. So when you're receiving data from the lab, they used a lab template similar to how there were field templates. They're available on the website. Similar to the field templates, you're still having to correct for human errors, data entry errors. The way that the data is coming in from the lab is a series of worksheets in one large workbook for each matrix. So the mantas, for instance, that's a single workbook, but it has 90-some worksheets. So you'll have to sort of combine all that together. We would recommend not leaving that data in Excel for that processing. If you pull it out into something like R or Python or SQL or some other language that you're comfortable with, it allows you to have a paper trail that records exactly what edits you've made, what revisions you made to those revisions. It lets you maintain granularity even if you decide to have sort of meta groupings of different categories. And it also just makes the process of updating data a little more simple. The other thing that is part of this process of receiving data from the lab is there are some situations where we had to either create or eliminate records due to lab comments. We had a situation where we had one sample container that was only 1/4 of the particles inside were analyzed, and so we had to extrapolate particles for the rest of the container. We had some situations where there were comments that identified particles but not full records. And then there was also a situation where the lab template that you'll see online, it had places to enter pieces of information that related to field collection, but they weren't necessarily always filled out because it's not really something that the lab needed to know a whole lot about. It was more about confirming which sample that they were analyzing, and so it's an easier process for us to link it back up with that sample ID that was recorded in the field and populate one full record using the field collection information that we have in the database and then the analytical data from the lab. So once we have everything cleaned up and standardized, where is this data going? The first place is the California Natural Resource Agency's Open Data Portal. It partners with various agencies in California, Department of Fish and Wildlife, Department of Fire, Water Resources, Ocean Protection Council, and it allows data to be stored in all sorts of formats. So you can have CSVs, PDFs, just the links to an Esri API for geospatial data. Our data will be a series of CSVs and PDFs. And then data is accompanied by metadata that describes the data set and helps people discover it. The Environmental Data Initiative is a similar portal process with a wide variety of options to submit your data in different formats. It also, however, issues what's called a Digital Object Identifier, or DOI, and the point of that is that it's allowing your data to be tracked, cited, shared in a more robust manner than a normal URL. The idea is that it is a unique identifier, but it also will link to the data as opposed to something like an ISBN. And then we have CDIN. So it's California focused, but it is very tightly controlled in its format and what values you can have for different pieces of information. And it also has to be submitted in a singular aggregated format, so you can't submit data like per particle. Instead, you're submitting concentrations. And I'll go into more details on that in a little bit. So regardless of CDIN, we're having controlled vocabulary within our own project. And so things that we worked on standardizing, these are sort of in order of difficulty. We have the polymer or material type, and then the various broad categories that sit above each of those. Operational size fractions, color, morphological category. Something like polymer type is, orders of magnitude more difficult than something like morphological category. We went from 280-some original polymer types submitted, and we ended up with something like 49, but it required the help of the scientists and looking at the spectroscopy IDs and confirming the different levels of categorization, as opposed to cleaning up something like color, which I'll go over in the next slide. So with color, with this project, we didn't have that nice color key that they have moving forward, which is a great improvement. But so for just mantitral data, this is an example of all the different values that we saw. And then in the far left column, sort of what we ended up coalescing to. And so some of these are just typos, trailing spaces, capitalization, which is relatively easy to clean up. Then you also had a couple of situations where sort of non-color information was included in this field, the black mats and black mat contaminants. Those are referring to the field blank contamination that Diana and Chelsea talked about. So there were also several things that we didn't standardize. We didn't standardize lab dates, personnel, spectroscopy IDs. That's sort of on us. We should have provided an acceptable format for all the date fields that were going to be entered by lab analysis. And because we didn't, we received dates in sort of such a wide variety of formats that it wasn't worth the man hours to fix all of them. And sort of similarly with personnel, we didn't want to analyze the data based on lab personnel. When we needed to reach out to Chelsea's lab to ask questions, we had enough information to identify the personnel, but not enough, like it wasn't standardized enough in order to allow us to aggregate it or analyze it. Spectroscopy ID was limited to one field, and we didn't really standardize it. We did to a small extent to assist the polymer material type identification. It's an opportunity for the future where spectroscopy ID is something that maybe should be standardized, maybe over several fields, identifying, you know, which library, what percentage match it was, to allow later meta-analysis and just a better record of exactly what came off the instruments. The other thing we had to do in data processing is, you know, deciding how to incorporate blank data. The data portals are both flexible, and so we're submitting all of the blank data to those two sites. That will allow people to assess their needs and use the data appropriately for their needs. I know Rusty, for instance, had to blank subtract in order to make the data work for the modeling effort, though for our report and for CEDIN, we are flagging or qualifying field samples based off of a quantitative threshold rather than subtracting out values for the concentration. So what does making data available look like? For CEDIN, it means you're picking one single aggregation. So in our case, it's a concentration. For example, a count per volume of each of the morphological categories and polymer types combined together and then split out by sample event and size fraction. So that's a mouthful. What that looks like is you would have a count per liter of polyester fibers in a matrix called sample water particulate, the size range at a site, CB7, on a date, August 22nd, 2017. And then it also requires populating a whole bunch of other fields that are standardized for CEDIN, but that aren't necessarily important to fill out in the field because they're the same for everything. So we have a project code for all the samples in the project. We have protocol codes that identify the field sampling methods, things like location codes. For the mantitrolls, all of the locations are open water. It's not something that you don't want to create more work for a field crew by making them fill out a whole bunch of fields that are exactly the same for a single sample. CEDIN, however, is limited in that it's missing places to put certain pieces of information that you might find relevant. So there's no place to put color. There's no place to put the particle dimensions. We can't include, you know, an average length or width of the particles that are in the concentration. The two data portals allow us to submit particle-specific data, but they also allow us to submit metadata, abstracts for the data sets, a list of investigators and collaborators. It allows you to submit spatial and temporal boundaries, different tags and descriptions that allow people to more easily find the data. It also requires that you sort of describe each of the columns if you're submitting tabular data. CEDIN sort of already has those descriptions as part of their data format. The data portals are also like I said, particle-specific data, so they're not aggregated. So each individual record is not going to be blank corrected or flagged, and it is a challenge to submit sort of samples with no particles found at all. You could submit all of your records with a count field, but 99.99% of your records will have a count of 1, and then you'll have a handful with 0, and that's very easy for an end user to overlook. So it's just a challenge that Sediment handles more elegantly in that when you have a concentration, you can have a non-detect that's more obvious. The bonus is you can include any piece of information you want, so there's no limits as far as we'll be able to include color and dimension of each particle, for instance. The data will be available in spring of this year, and that's all I have. So I will open it up to questions for the group.
Yeah, so for this section, we have been receiving questions, and so we're all here available for questions. So feel free to continue sending questions to this email address. So in a minute, we'll have a slide with where we'll post the questions so that we can all see the questions on the screen. Okay, so first question that we'll answer, there was a question on sieve size. Did you try collecting samples with different sieves? And if you had used a smaller sieve size, what might your result have been? So for the matrices, we mentioned we used the sieve was the third of the micrometer, and for the wastewater and for the stormwater samples, we used 2 sets of sieves. We used the 355 micrometer sieve that kind of matched the matrices sieve size, and we also added the 125 micrometer sieve to capture a smaller size fraction. And we know from looking at other studies that if we had used an even smaller sieve, we probably would have captured more particles, and it seems like you get more particles as you try to capture smaller particles. Then there was a question on air deposition and modeling. Would Rusty please address aerial deposition in terms of future research needed for fate and transport of microplastics into and out of the bay? Is anything in development? Well, first I'll start off by saying in the study we did not measure air deposition, and that is something that we hope to do in future research.
Yeah, and this is Rusty. I would just like to add, yeah, I think air deposition is a great question, but I think it extends far beyond just the modeling effort. And I would not say that we had enough data to really say from the modeling side that air deposition was clearly missing or clearly not a factor. So I think we just need significantly more field data, both in general, but also specific to the air deposition question, before we can really meaningfully incorporate that into the models and really be able to say anything.
A question on laboratory analysis. Could you elaborate on any tips or strategies for picking very small particles, less than 100 micrometers, and transferring to a Petri dish? We are having some trouble picking up very small particles and then confidently transferring the particle for analysis. Chelsea, can you answer that question?
Yeah, I'm going to hand it over to Keenan. Hi. So our tips for picking up really small particles, less than 100 microns, are, first of all, only do this if you're comfortable with it. So if you have good manual dexterity and lots of practice, then attempt to do this. But you might want to practice with other particles if you haven't done it before, because those particles are really hard to pick up. They're very small. So if you are picking them up, we use 4 sets. I usually have 2 pairs of tweezers or 4 sets with me at a time. We wet pick a lot of samples, so we'll pick spoonfuls of water with plastics in them. It helps a little bit to have water because you avoid the static issue.
So if you have one pair of forceps, you can pick up like a little droplet of water on one side of the forceps and then touch a piece of plastic with a droplet of water rather than actually attempting to pick up the plastic with both sides of the forceps. You can use the second pair of tweezers or forceps to scrape the droplet of water off of the forceps and onto the tape or whatever you're trying to mount on. Aside from that, we use wide-based microscopes so that it's really easy to slide from your counting dish to your sample prep dish rather than having to like stop, put things down, pick them up, and move them. That's it.
Great, thanks. A question on sediment remediation. Are there any large-scale separation technologies to remove microplastics from sediments for remediation treatment purposes? What is the largest volume of sediment that has been processed?
I'll take that one. This is Chelsea. And I want to kind of start by saying no, but I know that the Tijuana Estuary Research Reserve in San Diego, they were trying this. So they have a lot of plastic that comes down the Tijuana River into the estuary, and it fills up in the sediment. And they create these large sediment mounds that they've moved. So, I mean, this is a lot of sediment, like a small hill or mountain. And what they're doing is kind of sieving it, but through pretty large sieves. So I think it's maybe down to 5 millimeter or 10 millimeter, but they were trying to get the microplastic out of it because what they do with that sediment is spread it on the beach. So they'd be someone to talk to about their lessons learned. But I think large-scale technologies for this and sediment remediation for microplastics at this point, I think are pretty novel and new and haven't really been looked at in great detail.
Question on rain gardens. How did you go about sampling and quantifying effectiveness of rain garden nature infrastructure in removing microplastics? Here we have a citation for a manuscript where we described how we looked at the effectiveness of removing not only microplastics but other contaminants from a rain garden, as well as the results. So just briefly, what we did was we were monitoring a rain garden here in California, and during a storm event that met our threshold, we sampled water going into the rain garden at the inlet, and the water being treated by the rain garden flowed through an underdrain, and we were able to sample water coming out of the rain garden. And we monitored this through 3 separate storms, and we could measure the concentrations at the inlet and outlet. This was a project that we also worked with Chelsea Rockman on. And from the concentration differences, we saw that the rain garden removed over 90% of the microparticles through each of the 3 storm events. Next question, are there California-based analytical lab researchers able to do this type of analysis?
Does this mean commercial?
So the Southern California Coastal Water Research Project, or SCORP, is developing their capabilities to do this sorts of research, and as part of that, they are working with Chelsea to do a interlab comparison study to make sure that all the labs can come up with the same types of data when given the same types of samples. And many of the labs participating in that study are also in California, and it's a variety of different types of labs. So not just academic labs, but also various other government agency labs and more consulting firm type labs as well. Chelsea, do you have more to add about that?
No, I think that was great.
Next question. This data presentation seems like it could be a whole publication. Is there a plan for that? Yes. So we have several manuscripts that are being planned for publishing both the results as well as our lessons learned, and another one for the transport model. And so those are in progress.
Thank you.
Thank you.
Were there any more questions?
Oh, I think, is it not revising? Refresh it. Yeah, refresh it.
Oh, okay.
So
maybe I can, Let me
I think you jumped.
yeah.
A couple of
What number should I go to?
7, yeah, 7.
Okay. Question, how will the data be incorporated into Sediment? I am not aware of their current standards for microplastics. Seems novel and very important. Tony Hale, Dharma, and Wynn Calgar are working on updating Sediment to be more useful for trash data and would love to chat. Great. Adam, do you want to say anything on this?
So what I can say is that that same rain garden project that we were actually just talking about is data that's already in Sediment, or at least in Sediment format in our SFEI's regional data center. I think it's on Sediment as well. But I will talk to Tony. There definitely are some caveats on sort of what microplastic data can do in Sediment's current format. I know they're working on sort of Sediment 2.0, and I'm sure that there will be a lot of improvements to how they can handle sort of more novel parameters like macro and micro trash.
Great. And then I want to point out that Wynn is sharing an event that is happening next week. Were air blanks completed in the field or lab? Okay, so I think this question is about field blanks and lab blanks, and we did collect field blanks. I described earlier how we collected them. Most of the times the field blanks where we had another set of sieves in the field, and it collected any kind of air deposition that might have been happening while we were collecting the samples. And Chelsea's group also collected lab samples for their analysis. Chelsea, do you want to add to that?
I think the only thing I would add is this question specifically asks about air blanks. So instead of like putting petri dishes out to collect dust in the air from our lab, the way we did our blanks was we ran a sample that was similar to our samples through the whole process of the sample. So, for example, we would put RO water through the entire process of extraction, counting, sorting, all of that. And so we've done air blanks basically to test how well our HEPA filter was performing and things like that. But I think the best method for really being able to compare apples to apples with your samples are representative sample blanks and field blanks.
Thank you. Oh, I think that is all the questions that we have received. I guess we can end here. Thank you so much for all of your participation. We were really excited to get so many people interested in participating in our webinar. And just to remind everyone, the webinar has been recorded, and we will post it on our project website, and it will be available to everyone, so you don't have to be registered for this event to view the posted webinar.